Welcome everyone to Wednesday night at the lab. My name is Liz Jesse and I'm a Science Outreach Specialist at UW-Madison Biotechnology Center. On behalf of the Biotech Center, UW Extension and Cooperative Extension, Wisconsin Public Television, the Wisconsin Alumni Association, and the UW-Wisconsin Science Alliance, I would like to thank you for coming out to Wednesday night at the lab. We do this every Wednesday, 50 times a year. Tonight, it's actually my pleasure to introduce one of my coworkers, Dustin Rubinstein. Dustin is the director of the Genome Editing and Animal Models Core Facility at the UW-Madison Biotechnology Center, right here in this building on the 3rd floor, a couple stories up. He was recently interviewed by Isthmus, Business Insider, and Nova News. He grew up in the Chicago suburbs and went to the University of Illinois, Champaign-Urbana for his undergrad. He received his PhD from Cornell in 2008, where he also did a postdoc. Dustin has extensive experience in genetics applied towards a diverse array of research topics, including neuroscience, behavior, reproduction, and bioinformatics. He's always had an interest in generating tools to best address important research questions, like how the brain controls social behavior in fruit flies. Dustin was recently, that's kind of interesting, Dustin, you gotta talk about that a little bit. Okay. Sorry. Dustin was recently a member of a team of 3 Drosophila labs that worked together to publish the first CRISPR-Cas9 Drosophila paper. He helped generate a dozen genome-edited fruit flies. So tonight, Dustin will discuss the tools that were available before the CRISPR revolution, how CRISPR is used today, and how we hope CRISPR will shape the world to come. So ladies and gentlemen, please join me in welcoming Dustin Rubinstein.
Awesome. Well, thank you, Liz. Thanks for that excellent introduction. It's always fun to come out here and speak to Wednesday night at the lab. It's a great group. Thanks to all the partners, the laundry list of partners that you mentioned that put it together. So thanks to them. It's great to be out here and talk to you guys. So obviously I want to talk to you guys about CRISPR. We live in a tremendously exciting time where there's a transformative time in biotechnology where the ability to read genomes and write genomes and all kinds of synthetic DNA are all coming together to do some really exciting things. Maybe let's talk a little bit about maybe where that might lead us. So as Liz mentioned, I'm the director of the Genome Editing CRISPR core here at the Biotechnology Center. So whenever I like to talk about genome editing, I always want to make sure we start with the past, start with the history, because it's kind of hard to know what's so exciting about the era we're living in now if you don't really understand the hard slog it took to get there and what the techniques were before we had CRISPR. So I'll spend some time talking about how were we doing genome editing, loosely speaking genome editing. How is it being applied now in really tremendously exciting ways right now, like I should probably get back to work. But also, what are the really exciting ways in which we hope to apply CRISPR in the future, and how could it potentially transform the way in which we live? So sometimes I like to show this slide, and I know a lot of you people are going to look at this and be like, oh, okay, I know the difference between DNA and RNA and protein, but we're going to be talking a lot about kind of fluidly moving between these 3 things. So just to kind of make sure we're all on the same page, DNA is kind of like a menu. If you go to a restaurant, DNA is kind of like menu. It's got sort of a list of things that are there. There's a stack of them. There are a bunch of them. All on their own, the menu doesn't really do anything, but it basically, it lets you know the kinds of things you can order. And maybe you look on there and you see a cheeseburger and you're like, yes, that's what I want. When you get to the RNA stage, talking about activating those genes, that's kind of like placing your order. So that's kind of like having, the order made, and there are going to be some details in there. Maybe you want no onion, maybe you want extra pickles. But at the end, the real important thing, the real star of the show is the food that you get back. That's like the protein. So that's sort of the canonical way, we think about going DNA to RNA to protein. At the end, the protein's really doing the work, but the DNA is really important because it's sort of the recipe to make that protein. So sometimes I kind of like to show that. So let's start from the beginning of genetic engineering. When I say beginning, I mean beginning. Let's go way back. So if we look back, let's say thousands and thousands of years, approximately 10,000 BC, there were strains of wheat that were available. These are just wild grass strains. They look like the one on the left here, and you can see that they had nutrition, but it took a lot of work to extract that nutrition, and the amount of nutrition they had per unit of per grain was low. This wasn't the most desirable product. Now, over time, of course, through the process of artificial selection, you could see that these strains of wheat are leading to more and more delicious-looking strains. And you go all the way to the one on the right now, because of these giant wheat berries that can be mashed into flour and create lovely, delicious breads. So the strain on the right would be the bread flour that we all come to know. Okay, well that's great. We all know about artificial selection. Well, how could that relate to genetic engineering? Well, I like to show this case because it's actually really powerful, dramatic genetic engineering. So if we look at the original wheat strains, they were diploids. That means they had 2 versions of every chromosome. Now, over time, through artificial selection, there are these more delicious strains that actually led to a genome duplication. So now they didn't just have 2 versions, they were tetraploid. They actually had 4 versions of every chromosome. And even more, because 4 is not enough, why not 4 when you can have 6? So then there was another genome event that occurred that led to the strain that we think of as bread flour now, where there's six chromosomes. So these are hexaploid species. So my message here is that genetic engineering, when it comes to, in this case, agriculture, isn't something new. It's been something we've been practicing for thousands and thousands of years. And in fact, it's not like these were minor changes. What's more of a sledgehammer approach than tripling the number of chromosomes you have in a cell? These were really, ground-shaking, these are really powerful genetic changes that were occurring. So the challenge that we face moving forward from 10,000 years ago, is not to do more powerful changes, but to do more precise changes. And that's really going to be kind of the thread of how this technology moves forward. And of course, the stories of artificial selection. It's not just doesn't just lead to croissants, of course. Although that would be enough, right? All kinds of canine strains that blow my mind. That these are all derived from a recent common ancestor. Teosinte, the original corn strain, and the corn, of course, it's Wisconsin in the summer, you got to show a bunch of corn, and that's a hot topic of study here at the University of Madison, as well as this always blows my mind that the wild mustard strain has led to cauliflower, broccoli, and a number of other important food crops, of course, all through, right, isn't that crazy? So artificial selection is really powerful, and I think that kind of represents sort of the first epoch of genetic engineering. And of course, if you guys have any questions, feel free to shout them out. I like questions. And there's always bound to be some that are going to stump me. So I want to make sure I sound smart, so don't ask too hard of questions. Just kidding. Okay, so we're going to really skip forward. We're way in the past. We're going to move forward through our understanding of DNA and evolution and all these things. And we're going to get to what I would consider sort of the second epoch of genetic engineering, and that is the ability now to physically manipulate DNA. So the first really important study was done in Paul Berg's lab, taking a piece of the simian virus, the simian virus genome, and taking a little snippet of this lambda genome that affects E. coli strains. One of the big workhorses of labs, this lambda virus affects E. coli. They were able to take a little tiny piece of this viral DNA using molecular scissors that at the time were really exciting. So these would be restriction enzymes, right? They recognize a few sequences and they can physically cut DNA. And then pairing that with basically molecular glue would be ligases. So this is really transformative. So the ability to not just look at DNA and not just see how it's arranged and be like, oh, so it's a double helix, and not just know the sequence, but now this is the first time there was actually the ability to actually manipulate it, right? Cut it where you want. Loosely, cut it where you want. You're kind of limited by your options, but cut it where you want, and then stick the pieces back together. And that was a really important age in genetic engineering because it led to all kinds of really exciting things. And of course, the extension, not just taking one virus genome and putting another, but another obvious case. And a few years later, another lab took a piece from the really important early embryo developmental model, Xenopus, the African toad. African claw-toed frog, I think. A snippet of the genome was cut out and cloned into the E. coli genome. To show that this isn't just limited to viral genomes, but DNA is DNA, and you can take DNA from all kinds of species and put them into new species. And that's all great. This was, I think this paper was in 1973 or 74 or so. I should have the time frame up there to better understand that. But it, as exciting as this was, it also raised a pretty concerning red flag. Well, what are the implications of this? Are we going to be making, animal-human hybrids? If we can stitch DNA together, we've all seen science fiction. We love science fiction. What are the ways in which this could go wrong? So there as important as this work was, and led to Nobel Prizes, of course, there was a broad concern that perhaps we really need to evaluate what the impacts of this technology might be. So there was somehow they got all the scientists to agree to put down their pipettes, put down their work, and fly out to California to Asilomar, this lovely beach location, to come together and halt research along these lines, and come together. There were, I think, almost 200 attendees, and that included a lot of basic research scientists, lawyers, physicians, people coming together and trying to understand, well, what is it that this technology has to offer? What are the concerns and the risks? And I think it's actually, I think it's a great moment in sort of science history, because the community as a whole was able to come together and publish this document that, in a sense, still drives the biosafety protocols that that the NIH and many other bodies demand today. So I think that's kind of one interesting lesson going forward. And of course, I'd show you a piece of frog DNA being turned into bacterial DNA. Well, that exact same idea led to a number of other really exciting things. For example, transgenics. So now we're talking about the ability, remember, to manipulate DNA. So you can take a piece of DNA, you could maybe stick two different pieces of DNA together. Maybe there's a gene of interest you want, and maybe you could take a fluorescent green fluorescent protein and stick it on there, so you could follow the protein around. You could take a piece of DNA, for example, and stick it into stick it into rice to provide more nutritious rice strain, or resistance from a number of insect pests, or for, here in this case you could see mice that are glowing red and green. So here, what this sort of second era of genetic engineering gave us was the ability to manipulate DNA. So you could take this DNA and essentially stick it into the genome, and it would sort of hop in randomly. It would integrate into a random place. So it was great that you had the ability to manipulate DNA, but where it landed was sort of out of your control. So you might have this mouse that has this red fluorescent protein, and that's great and that's really powerful, but what if that gene happened to land into an oncogene that causes cancer? Now, you might think, oh, when they express red fluorescent protein, they get cancer, eureka. Well, no, it might be because they landed into that gene, or it might be, my point is there are a number of complications if you can't control the exact place that gene goes into. There's a lot that you can't really know. So while what transgenics gave us is the ability to build pieces of DNA that are really useful, what it didn't allow us to do is at the genome sequence level. Let's say if you have this gene of interest here, this is your gene. It didn't allow you to go into the genome and say, I want to focus on this gene, and I want to change this gene specifically in just this spot and exactly know where I'm at. So what are some things that you could do with that? Well, one obvious case is if you're just interested in genetics, you want to know how genetic networks function. The first thing you could do is, well, knock the gene out and ask, what's the effect? Do mitochondrial function change? Does stroke risk get alleviated in an animal model? Another really exciting thing, another important thing that you could do, for example, is to, as I mentioned, maybe take your gene that you're interested in, right in the spot where it lives, and tag it with something that you can visualize, like, again, green fluorescent protein. So you can follow it around the cell, you can stalk it and see where it goes and see who its friends are, and you can grab that protein back and see who it's interacting with, and you can get a sense of gene networks that way. and also edit the sequence itself, and I'll talk a little bit more about how that's useful in a bit. So okay, so that's great. So what I'm kind of getting into is what's this sort of third era of genome engineering or genome editing, where you're actually going to the spot that you want to edit and change it. And the way I often like to describe it is we live in a really exciting era of bioinformatics, right? Where there are just genomes being sequenced left and right? I mean, we have problems in how to manage these giant data sets, and I couldn't even tell you how many nucleotides of sequence are known. And that's great. Being able to read those genomes is incredibly powerful. But I think the way that, at least kind of the way I kind of think of it, often when you read these genomes, it's kind of like if you have a genome, it's sort of like printing it out and having it as an encyclopedia. You want to know how this gene that's important in colorectal cancer is spelled. Well, then you flip to page 26 and you see, well, in this person it's spelled this way, and in this person the gene is spelled this way. And you might think, well, in this group of patients, they have much more high prevalence of colorectal cancer, for example, and they tend to have a spelling of this paragraph spelled this way. So you might think, oh, well, that could be really exciting, but that's really just a correlation, right? You're just kind of flipping through the pages. What CRISPR allows us to do is basically, it's more like opening up in a word processor, opening up in Word. So now you can go in there and you can actually go through and punch in the new sequence and change it and ask, how does that affect the phenotype? How does that affect the effect that you're interested in? You can go through and change the spelling and ask, now, does this mouse get cancer, for example? So how do you do all that? So before CRISPR, there are several other strategies, and it's been known in a nutshell how you could do it. And basically, if you can create something as simple as just a break, well, I often will call it just a double-stranded break. I'm probably going to say that, but all I'm talking about is just a break in the DNA. If you can just get it to break the DNA in exactly the spot you're interested in, you can create a genome editing hotspot. And it's not in the way that I talked about transgenics, where you're kind of cutting one piece to stick another piece in there. The strategy is actually different than that. The nucleus of a cell hates broken DNA, and that's like a 3-alarm fire. When it recognizes a break in the DNA, there are tons of different pathways that get recruited, and tons of things that are holding on to different pieces of broken DNA, and all kinds of different proteins showing up and asking, all right, well, what's this supposed to be? How do we fix it? So what you can do then is, by generating a break in the DNA, is use the cell's own ability to fix that DNA against itself. So while these things are normally there to sort of fix it, if you can kind of hijack that system and fix it in a way that gives it the change that you're looking for, then you're in good shape. And I'll give you kind of 2 examples of roughly, oh yeah, go ahead.
So is breaking DNA the same as cutting DNA?
Yeah, in this sense, yeah, I'm just talking about a cut. Yep, so just this.
How do you cut DNA?
That's if you asked me that question 3 years ago and I had the answer, I would be a billionaire many times over. So, and that's exactly the challenge. How do you cut DNA in exactly, because remember, as I was saying, the challenge is always about precision, about not just cut it, but cut it in exactly the one spot where you want and nowhere else. And that's essentially the question. If the title of my talk had been, how do you cut DNA? It would maybe not have been as exciting, but you exactly hit on the essence of what CRISPR is. So we'll get to that. I'm going to pause, but that's a great question. So you have broken DNA, and I mentioned there are all these different pathways to fix it. So there's basically kind of 2 different things that can happen when you cut the DNA. And these are different proteins, different people, I'm anthropomorphizing, different proteins and different things that show up to the scene to fix it in different ways. And one is sort of an imperfect repair. So they got sort of 2 broken ends, and sort of make their best guess and stick the 2 ends back together. And maybe they were right, maybe there might be a couple nucleotides missing here and there in the process. But you can imagine, if you're familiar with the way that proteins are encoded in the genome, a deletion of a single base changes the whole protein coding sequence, right? So even a deletion of a single base in a gene here, you're essentially going to knock out, you're going to break the entire gene. So that's one strategy. Sort of just to cut it and have it imperfectly be repaired. Another is a perfect repair strategy. So in this case, the strategy is, well, we're going to cut it, And then the cell's going to freak out. And it's going to ask, well, what is the sequence supposed to be? And presumably it's probably looking for the other chromosome, right? We're diploid. We have 2 versions of every chromosome. So, you can imagine that it's probably asking, okay, well, what's that guy doing over there? What's that sequence supposed to be? So if you provide a template to repair where there's similarity here and similarity there, it's going to think, oh, this is the sequence it was supposed to be. So in this case, sorry for the jargon, but let's say there's a red, you can change the DNA sequence kind of by tricking it into thinking what it was supposed to be. Does that make sense? So these are kind of basically the 2 sort of genome editing strategies. One where you're kind of cutting and the cell sort of hopes for the best, and then the other one where you're repairing and changing the sequence exactly how you want it. Now you might ask, well, why not just always do this and change it to what you want? The challenges in general with genome editing is that the cell always prefers this way. And it's one of the big challenges with CRISPR as to how to push things towards this, how to get it to occur more towards perfect repair. And we'll get to that later, but those are kind of the two methods. So I mentioned that, we've known for a while that breaking the DNA is really the important thing. And CRISPR wasn't the first platform that was developed to generate those cuts. So one example here are zinc finger nucleases, where there was you could generate synthetically a protein that would find the sequence of the genome you're interested in. So you'd say, okay, I'm going to design a whole protein that's going to recognize this sequence, and then I'm going to design a whole another protein that recognizes that sequence, and then they're each going to have these molecular scissors attached to them, so they'll each cut, where we want. And that's fine. So, those are zinc finger nucleases. Another variant improvement on that were these talon nucleases. And again, the same sort of thing. You're going to generate this giant protein that recognizes the bases you want. And that worked. You create a double-stranded break in the sequence you're interested in. But yeah.
How do you know what kind of protein you're making when you create a synthetic protein?
That's a really good question. So how do you know it's right? So you're making it, you'd be like, how do you know I didn't make a mistake?
Repeat the question.
Right. So she asked, when you're making a synthetic protein, how do you know it is what you think it is? How do you know you're making the right one? And you're kind of hitting on a really important point that creating these proteins is actually really hard. It turns out that creating a protein is just much harder. It's harder to know that you did it right. It's harder to know that the sequence is what you think it is. It takes a lot more money. It takes a lot more time. And it's way more expensive to make a protein sequence than it is, for example, to make a DNA sequence. So you could do things like you could sequence the DNA that encodes for the protein, and you could know that way. There are a number of other methods. That would probably be the easiest ways to sequence the DNA that is the recipe for that protein. But even that takes a lot of resources. So there'd be a huge benefit if instead of generating a custom protein every time, and these could cost $20,000. This could take a couple of years, and to put that in perspective, that would mean that if you're a lab and you really think that you found an important gene that might be important in cancer, you better not be wrong because you've sunk a lot of resources into that gene, right? So into knocking out that gene. So it was a challenge. It could be done, but it was a challenge.
What's the significance of some capital and some small letters?
I'm not sure. Probably.
Like a dominant gene or something?
Oh, it could be, right? So I know a lot of times, sometimes some genes, you see the capital letters are dominant and the little letters are recessive. You're right. I mean, that is a really common way to. Very smart.
The recognition sequence.
It might be that the capital letters are the recognition sequence here, and the lowercase ones aren't. So it might just be telling you that this protein is designed against those capital letters. But I'm not sure in this case exactly. So that's zinc fingers and talons. But now we're going to really start to skip into CRISPR. But before I do that, I want to kind of take a little step back again and talk about really briefly about kind of how we got to where we are with CRISPR. So it all started with the first observation when someone was looking at so this was in 1987, so there weren't a lot of DNA sequences around, but there was a gene that they were interested in, and they happened to notice in this place, this piece of DNA, there's a gene they were interested in, this bacterial genome, and next door to it, not where they were really meaning to look, but it was kind of weird because there were these sequences, these purple sequences that were repeating. And at the time, that was just kind of like, oh, that's kind of strange, and it got published. It was noticed more and more and more, and because scientists do all kinds of research and not just think about how is sequencing this gene in E. coli going to cure cancer, because there are people just wanting to know how does this E. coli work. People published and exchanged information. And eventually, if we skip forward up to 2007, it was realized that these sequences, over time throughout the early 2000s, it was realized that these sequences in between the purple, these repeating sequences were pretty cool. I mean, that's kind of a strange, it's kind of a but as more and more DNA sequencing data became available throughout the late 90s and the early thousands, people started to actually cut out, look at those gray bars and those black bars and those light gray bars and ask, okay, well, what's that stuff in between? And one of the really wild things was that those sequences matched viral genomes, right? So we're talking about bacteria. And bacteria live in a really hostile environment where they're viruses, in this case. It's kind of scary. It looks like a zombie movie or something. But there are all kinds of viruses lined up to chow down on this bacteria. So the thought was that these sequences in between were actually part of the adaptive immune system. So this is an E. coli, version of an antibody. So how would that work? So for example, what we know does happen was that a phage, so a virus that attacks bacteria, would inject its DNA in. The bacteria would recognize this as being a foreign piece of DNA. It would chop it up into little bits. It would take those bits and store it in its own genome as sort of a memory, as sort of like a fingerprint. Like if you ever see this guy again, you ever see this sequence, this guy's trouble. So then how do you use it? That's how you store it. And how you use it, you could transcribe that region, now cut those bits up again, and now you basically have a bunch of kind of wanted posters posted. So if you could look for the little blue piece, if you ever, so that there would basically be little sentinels, proteins carrying these little pieces of CRISPR RNA that would sort of wander around the cell, and if it ever found a DNA sequence that matched that blue sequence, which is essentially a memory of a previous infection it had, it would chop up that DNA and chop it up so much it never had to worry about it again. So that was basically the idea, is the function that CRISPR served in bacterial cells. And the reason I think it's really important to talk about this is not just because it's really cool. I mean, basically that bacteria and archaea, virtually all bacteria, have a memory of their infections, which is crazy, and basically have their own version of antibodies. I mean, that's pretty interesting. But also because I think it's really important, this is going to lead to CRISPR, that this whole system came from not people sitting in their lab trying to cure every single disease in the world and not trying to specifically ameliorate one problem with humanity, but because people just wanted to know how biology worked. What are the basic principles that drive biology, that drive an E. coli cell versus a mouse cell? So really, this is, if there's nothing else, CRISPR is a triumph of just basic research, of wanting to know, figuring out an answer, and then clever people all throughout the way, but turning it into really important research tools that hopefully will change the world in which we live. That got kind of trimmed. So if you remember, so here I was telling you about, I spent a while telling you about how the really important thing you need to do is if you can cut the DNA exactly where you want, you can basically do genome editing, right? That's the secret sauce right there. So what was discovered here now is this protein that cuts DNA programmed by that little piece of RNA, right? Now these are now all these bits are kind of coming together as a tool that can be really useful. So In 2012 and 2013, there are these two different labs published the actual application of CRISPR to cut DNA exactly where they want. So what do you need to do it? So let's take a look here at how you might design a CRISPR experiment. So if this is your genome region here, and each bar is a nucleotide, we're going to throw out some jargon here. We have a target sequence right here. So this is 20 nucleotides. This is the part that you really want to get at. There's a PAM, a proximal adjacent motif. That you can't change. In this case, it just has to be GG. You know, there's ATCG. As long as it's GG, any target up here is available. So remember, I showed you that little CRISPR RNA. There were some blue ones, some purple ones. All you need to do is synthesize a piece of RNA that carries this sequence. And just as you were getting at, creating a protein in the right sequence is really difficult. It's really expensive, and that's what drives the cost of, I mentioned $20,000 and years and all these things. But creating an RNA is trivially simple. I mean, if I can go upstairs and start working on that, and by the end of the day, tomorrow or the next day, we'll have RNA ready to inject into mouse embryos and get right on it. And the cost of that is really small. So the power of so here, remember, so we're generating this guide RNA here, which finds the target sequence you're interested in, whatever gene you're interested in. And then we need this Cas9 protein. This is sort of the workhorse in that E. coli cell. This Cas9 protein recognizes all these little bits, and it says, okay, this is a foreign invading virus. I'm going to cut right in this spot, right here. And in that way, we can create double-stranded breaks. We can break the DNA exactly where we want. With a nice, really specific sequence, 20 nucleotides, you're not, in general, going to find that sequence happening a lot elsewhere. So we're really talking about one spot in the genome. But what's even more important now is that these are it's basically, and you can program it as to where you want to cut it, but it's like dirt cheap to tell it where to cut it, right? So before I was saying, if you have a gene you want to target, it might be 5-10 years worth of research of a lab. It might be, you might have 4 or 5 people working in your lab, and it might be, 40-50 human years worth of work before you felt confident enough to say, we're going to knock this gene out. Now, since this is so cheap, we can go through and we can screen, so we can target tons of different genes, or this can be sort of a side project. So, and it works really efficiently, and that's one of the big draws of CRISPR, and we'll talk about what all that stuff can, what that can get you in a bit. Any questions so far? That's sort of that's kind of the big meaty slide. So, anything. So yeah, so CRISPR creates breaks in double-stranded DNA exactly in the spot we want. It's super cheap compared to previous methods, and it's being used all the time now in labs throughout laboratories throughout the world. And I'm showing this slide again because this is really how it's being used on a regular basis to knock genes out, to disrupt gene networks by knocking genes out, to add to decorate your proteins to see where they go or whatever.
Can this be used can bacteria or good bacteria or viruses, possibly with edited genomes, do this work?
That's really interesting. So having bacteria kind of do the heavy lifting for us, create little bioengineering machines. Okay, well, I had that idea first. So before she tries to patent, I think we can all agree that I said that before she did. Well, no, I think ideas like that's real. I mean, that's a really good idea. So how can we? So you're once that happened. You're way past my slides. You're so many steps ahead of me. So how can we have a system like this, right? How can we make it as powerful as possible? And E. coli are really great workhorses of cells, and that might be one way in which we could use it. That's certainly possible. I can't think of how that's being used now. So if the IP people, if our lawyers are listening, they're going to want to talk to you afterwards. But But I think ideas like this is really exciting, is that it's kind of wide open, and we're going to need a lot of people with really good ideas to figure out how to use this stuff. So don't go too far. So I talked about how efficiently it works, how cheap it is, but also I think one of the really important things is it works, how universal this platform is. So it used to be if you wanted to do genome editing, if you wanted to knock out a gene, you basically had to do it in mice, maybe fruit flies, and maybe a plant, one or two plant species. But now virtually it works in just about every species in which it's been tried. The whole, everything from rabbits, mice, rats. We have it now working in. We work on pigs here now. Basically all the E-I-E-I-O animals you can think of. And of course, the really tried and true, my love, the fruit fly, and a number of crop strains, as well as cell lines too. So these could be human cell lines. So you could take a patient's tumor cells, and you could edit the genome of those tumor cells and ask, well, does it act less like a tumor now, right? So did I hit upon its, that gene that makes it be a tumor, for example? So there's really, it's not only that it works and it's cheap, but it works in tons and tons of different platforms, tons of different species and tons of different applications. And that's that's really exciting. Hopefully you guys are all on board that CRISPR is pretty exciting. But there are, of course, some wet blanket moments, so there are some drawbacks, right? So the most important thing when you're editing a genome and you're doing the research that follows it up, you think you made the change, and you're measuring all these variables and checking all these things, and then, because science is all about tracing causality when you can. You made this change and you want to see what's the effect. Did it cause these effects? Well, what if CRISPR wasn't as specific as you had hoped and it made a change somewhere else, and all the changes that you thought were due to the change that you identified were actually due to some other change in a different gene, right? Then you'd be misascribing the function of this one change, where really it's this other thing over here. And that's one way to put it. And think about it now if this is a human therapeutic, right? Now what if I tell you, well, I'm going to change this, I'm going to fix this one gene of yours, mostly. I might also change some of your other genes, but I think I could probably fix that one gene, and I think some of the other ones might be okay, right? So if you make more changes than you think, it's a big problem. So the ability to be specific and make the change exactly where you want it is really important. Go ahead. Yeah.
I'm just curious. How do you spread it throughout the whole body?
Yes. Yeah, and that's a great question. You're asking all the smart questions. You're stealing all my. If you have my slides, can you see my? and we'll get to that. We'll get to how, because you can imagine if you have a problem with your lungs, how do you not make changes in your heart and make sure that your lungs get the change? And that is a big challenge. And we'll talk about a few different ways in which we can hopefully use it for medicine for exactly that reason. Because good question again. Yeah.
So CRISPR is more precise than earlier ways of editing.
Yeah.
But it's still not quite as precise as we would hope.
Good. Yeah. So I should finish that thought. So CRISPR is pretty precise. Depending on who you talk to and how you're using it, it's maybe a little less precise. The lesson, so early on, of course, the obvious question is, well, are the changes, just to restate what I was saying before, that there's a big problem with off-targets. So that was sort of the obvious question. So what are the answers? How do you address that? Well, it became pretty clear that there are a couple possibilities. So here's an example here, right? So you have this sequence here. It's exactly where you want to cut it. Now, what happens if there's another sequence that exists on that green chromosome over there. It's just off by a single base pair. Is it still going to cut? Well, it turns out that one of the most useful things you can do is just do your homework ahead of time. If there's a sequence that you're targeting that has a bunch of similar but kind of difference all throughout the genome, don't use that one, right? So be smart from the beginning and be really careful about your design. So that's kind of one approach. Another, there's tons and tons of other, a lot of work being invested into how to maximize specificity, how to minimize, how to make sure that it only cuts here and refuses to cut here. So that's a really active area of development. So CRISPR is, it's not massively more specific, but these are really important things to consider when you're using it. Oh, yeah, sorry.
So, sorry for the silly question.
No.
But so far we've been talking about how much we've advanced being able to manipulate the DNA of cells.
Uh-huh.
And part of the manipulation process apparently is building what's called gRNA,
Right.
RNA.
Yeah, okay.
I don't think that's a protein.
It's an RNA model. Yeah, you're right. Sorry.
Find what it was earlier.
Yeah.
So how do you change or how do you build gRNA? How do you build those sequences to match the
Right.
DNA that you want to change?
Yeah, and that's a great question. So it turns out making, basically, just like the way that you make DNA and you turn on a gene, you can make RNA. You can either make a piece of DNA and transcribe that piece of DNA to get a piece of RNA. Are we on board? Am I getting too jargony?
No.
So you can get RNA from a DNA sequence, and people have been making DNA now for so long. There are companies that have it worked out to make small pieces of RNA. So that guide RNA is only 100 base pairs. Now, I mean, is that a lot? Is it a little? I can tell you it's really short. 100 base pairs. It's trivially easy to make a piece of DNA. That would cost a piece of DNA, 100 base pairs. It would cost you, something in the order of $100 or so. But you can do some tricks to make DNA. You only really need to make this target sequence, and that's like $4 or $5 to make that. And then you just have to basically turn this gene on and express that gene, so to speak, and you just get tons and tons of this RNA. Now on the flip side, because synthetic biology and chemists that just blow my mind all the time, they're actually just straight up making these RNAs. So you just add one base, and then you wash out all, and then you add another base, and then you add another base, and you just do it one at a time. But they've got it worked out so that actually is a really cheap method as well. So hopefully that kind of answers your question. But the strength is that you're only making 100 base pairs here versus a protein that's going to be thousands and thousands of base pairs, and you can't really, each one of those is different. Whereas here it's only 20 nucleotides. You really only need to make this small part. The rest you already kind of have.
As you were saying earlier, with our current technology, we wouldn't be able, some sequences are so similar, we wouldn't be able to edit them.
Yeah.
Is it possible that we would be able to edit them in the future?
If they're exactly the same sequence, that's hard because we're all about tracing down its address is all about the sequence. That would be really hard. So hopefully what you look for is just maybe it's different enough but close by. Maybe there's just a small difference and it's just not as close as you'd like. That's the only, yeah.
So you couldn't change a single G to a C?
Yeah, so how could you do that but not change it in the other spot?
Right.
Yeah. So, and that is kind of a problem, for example. There are some genes that have been sort of copied. They're sort of copies existing in the genome, and you want to just knock out one version. And over time, evolution has made this one version maybe be important in cancer, for example, and this other gene is important in something else. So how do you just change the one without changing the other, even though they're really similar? That's one problem. Another is that fish, for example, have their genome is quadrupled, so they have 4 versions of every gene. So how do you just knock out the one? So I mean, these if we're basing everything by sequence, again, it's a really important question. If we're basing everything by sequence and the sequences aren't different, how do you really target it? So I think, yeah, I mean, I guess you'd want to find a difference nearby and kind of go with that. It's not really a satisfying answer, but.
So you just edit a larger sequence.
Yeah, or maybe edit it from farther away where there is a difference and kind of hope that you can sort of make a change way over there. Yeah, that's a really good question. So at this point, I kind of told you about how we're using it now, all the things that we are doing. I kind of want to transition now to how do we hope to use it in the future. It's going to be kind of biomedical heavy because that's sort of my background, but hopefully I'll kind of show you some other applications as well. So what's the hope for CRISPR and new therapeutics? So if we kind of think about all the challenges, all the ways in which we can use CRISPR and all the different challenges, we could kind of group them into 4 kind of general approaches. So the first is autologous immune enhancement therapy. I mean, it's basically, can you take cells and can you engineer them to do the things, can you take cells from a patient and engineer them to do the things you want them to do, right? So basically carry on maybe a different function or enhance their function. That's one possibility. Another is ex vivo, right? So you're taking out some of the disease cells, some of the cells that have that genetic dysfunction. And then, over here, while the patient is, healthy and going about their business and going to work and all these things, you can make the genome editing change here, and you can verify it, you can make sure it's right, and then you can put those cells back in the person, and then they'll be cured, so to speak. That's another approach. Another is in vivo therapy, right? Now, what if we're talking about muscle, for example? You can't be like, okay, I'm just going to let me borrow your, I don't know, your quadricep for a second. I'll be right back. You just sit tight, and then you fix the quadricep. So how is it that maybe we actually change the cells in the living body? And then finally, germline editing therapy, right? So now we're talking about actually changing the sequence of an egg or a sperm, right? Now, in that case, you're changing all the cells. You're changing all the cells and all of the cells from that one cell, so all the progeny of that person in perpetuity. So I've arranged these in kind of ways that these are, the top are sort of the most proximate. That top one is happening right now. There are multiple therapies occurring right now. And the bottom is sort of the one that's sort of most, the further away and has the most, obviously, kind of bioethical implications. Well, I'll give you an example of all of them.
That last one wasn't the last published paper involving HCM, I think it was,
yeah.
where they did just that. They
did.
took the sperm cell and then united the egg and the sperm in an in vitro situation.
That's right.
Yeah.
Yeah,
so
exactly.
The last one was done just recently.
It was done, but I guess what I'm saying is it's not a therapy. It was never meant to be a therapy. It was sort of a test. Yes, you're right. So let me rephrase. So in terms of as a therapeutic, so we know we can do all these things. We do all these things all the time. I mean, we can change, all these things, are possible in animal models or in a number of different applications. We do these things. We do germline editing therapy at the bottom, the most challenging one. I mean, that's my bread and butter. We do it all the time. The question is kind of how close, thanks for helping me refocus that, how close are these to an actual therapeutic where we'd be willing to look someone in the eye and say, we're going to heal you by doing this. And that's a challenge. So autologous immune enhancement therapy. So the goal in this case, kind of one of the cool things is that T cells can actually recognize certain forms of tumors. They have an innate ability to recognize certain tumor markers that occur. And there's basically a natural defense mechanism your body has for certain types of tumors. And so the goal is, can you basically bump up the activity of those tumor-seeking T cells? And can CRISPR be used for that? So using just sort of transgenic technologies, right? Not genome editing, but just sort of sticking in a piece of DNA into the cell, into T cells, were really promising. There are therapeutic trials for that. But does CRISPR allow it to work more efficiently? So just to kind of outline it, so we have the situation here. Let's say someone in a really late stage lung cancer, for example. You would be able to harvest their T cells. And this is a trial that's currently underway now at the University of Pennsylvania, and since then there's been another one at the university in China, and I can't remember that. I'll show you in a second. And this is now this therapy is actually underway as of, I don't know, somewhere around April or so. The idea here is there's sort of 3 things that are occurring. This is sort of a pre-CRISPR technology. So here they're basically adding in a version of the T cell receptor that recognizes markers that tend to show up in tumors. So you can basically make these T cells want to find tumors. But one of the tricky things why cancer can be so difficult is that T cells have this checkpoint system where basically they can basically be told just to stand down. And tumors, and this biology escapes me, somehow have a way to kind of hack into the T cells and have a way to turn on that checkpoint. So they can basically get them to stand down where they normally wouldn't. So this gene PD-1 is important for sort of repressing the tumor-seeking activity of these T cells. So the idea is to use CRISPR now to knock this PD-1 gene out, as well as knock out the normal T cell receptor gene. So basically these are just like laser-guided tumor weapons, right? That's the idea. So now you're knocking out the normal T cell receptor gene, and you've added in this one that's going to find tumor antigens, and hopefully that's going to be extremely effective. And then you would add those back to human patients. So there's also 2 trials now around the world. Another one in Sichuan University. So that's one case where you're basically, basically kind of bioengineering your own cells to be effective towards cancer in this case. So let's move on to ex vivo editing therapy, and this is also really exciting. So the case here of sickle cell disease, the genetics here are really nice and simple and convenient. A single point mutation, a single variant in this gene HBB, the 6th glutamate is changed to a valine. It's a single nucleotide that causes sickle cell disease. So the strategy here is, could you take hematopoietic cells? So these are cells, bone marrow cells, that can potentially, take on all the different blood cells. They can to generate all the different blood cells. So what if you could harvest their bone marrow cells? You could change it back to the sequence it's supposed to be. And then, hopefully the hope would be to add to basically graft those cells back into the human patient. You have your own bone marrow cells fixed, right? Now we're not talking about finding perfect matches. You're your own, what's a better match than yourself? So that's one of the ideas here. So you're taking out the cells, you're fixing them in a dish, you've confirmed they're fixed, and then you add them back and you're cured. So in terms of sort of proof of principle, it's worked really well in mice. So they could take these, add it back here in mice, and you can see that the sickle cell hemoglobin, HbS, is a lot lower here, which is good. And the HbA, which is the healthy version, is a lot higher here, which is also really good. So in this case, you can see that this therapy basically is curing the sickle cell disease in this mouse.
So,
sure.
Are you saying that with CRISPR, you just need to make the cuts, and then the cell is going to use either of those, the imperfect or the perfect repair method, like it was doing in previous models of gene editing? And if so, what happens if both copies of the chromosome are bad? You can cut them both. Yes. But how do you get any fix in there?
Yes.
I missed that part.
You're right. So, thanks, Isabel. So if you remember kind of way back, I showed those 2 branches where there's the imperfect repair pathway and then there's the perfect repair pathway. If I can recapitulate your question, how do you know that you're getting both perfect repair in both chromosomes? What if you just break it and make it worse?
Well, I guess my question is, if there is no good copy, what is the cell even doing?
How does it know what it's supposed to be?
Right.
Oh, I'm sorry, right. So in this case, okay, thank you, right. So whenever you do that perfect repair pathway, you're always giving it a template. So you're always providing, this is what the sequence is supposed to be. So in this case, you can actually use just a piece of DNA that carries the sequence that you want it to be. And it's going to find enough, it's going to be like, okay, well, if there's a cut right here, it's going to be like, well, this sequence is the same and this sequence is right, so whatever's in the middle here must be what I want. And what it then does is it makes that donor sequence the genome sequence, and it basically carries over that fixed version. So if you're hoping to have a perfect repair, you always need to give it a template to repair to. So sorry if I didn't make that clear, but yeah. So that always requires a donor. Thank you. What we call a donor sequence. Yeah.
So do those repaired cells regenerate themselves once they're in the body? I mean, there's a few cells in take care of all the
right.
marrow that is wrong?
Yeah. So okay. So that's a great question. I'll answer it. You guys are like, obviously you guys can see my slides. I don't know, but so, and it becomes a problem in the next slide. But you know what you could do in this case is you could do like irradiation or something. So you could deplete them of their bone marrow cells, right? So you're basically killing all their bone marrow cells, and you could graft this back in. So now you got a huge head start. So they're healed cells. And I believe, you know, I'm the farthest thing from the guy who would do that. I'm not a clinician, but I think that would be a strategy because you're right. So you know you're going to make the changes. But it's only going to be a few cells. You know, each one of us sitting here is, I don't know how many cells. Anyone know how many cells are in a body? Way more than I can edit efficiently. So how do you outcompete? So how do you need to know? So, and that is always a big challenge. So if you're going to make a change, either has to be like just 100% efficient and just all has to make the change you want, or there has to be some way that you can select for those cells. And when we get to this next case here, in vivo editing therapy, we'll see a strategy in which that sort of worked out. So Duchenne muscular dystrophy, a debilitating but unfortunately common disease. There's a case here where there's a mutation in an exon in the gene where instead of just having it normally chug along, it basically tells the protein making machinery to stop. So obviously that breaks the gene in a way. And now there's a mouse model for that gene. So now we're talking about this has been done in mice. So you can imagine one strategy, and it just so happens, let's say you can just kind of take my word for it, that little piece, number 23, you don't really need. It does more harm than good. Having it stop is way worse than having it there at all. So essentially, if you just cut out 23, the protein pretty much works okay. So what you could do, because this is muscular dystrophy, so these are now muscle cells, right? I mean, all your skeletal muscle cells. And we get to that challenge of how. Okay, so wait, back up. So what you could do now is you could basically make what they did is they loaded into a virus the ability to make these all the components you need to cut this little number 23 out. Injected it into a mouse tail vein, and got a sufficient number of genes of cells where this actually got cut out, and you can see, you can see that the muscles were able to generate force. So, and this gets at kind of both two questions. So, you were asking how do you change all this? How do you just change the cells you want? And you're saying, well, how do you outcompete them? Well, one of the things that they found is that they were able to fix the muscle fiber cells that are sort of progenitors for other muscle fibers. So these are so your muscles are kind of always being replaced, and there are these little muscle fiber precursors. And if they could change a few of those, then over time that could kind of repopulate. And you essentially get like an expanded effect if you could just change those. Yeah. Go ahead. Yeah, sorry.
Would it be more promising then in this area of research to kind of choose genetic diseases such as Huntington's disease, or ones that start later that you could have more time to address the changes that need to be done?
Sure. Yeah.
I'm thinking of ALS too. That
sure.
would be one where the expression is later.
Yeah.
But the awareness that we've been able to map this out, we know what genes could be involved in this, and we'd be able to address this earlier on. Yeah,
and that's a good point. So, and I think that certainly is going to be a really important strategy. How much of a if you're already in the throes of a disease, that's a problem. Right. And I think that's a really important factor to weigh. I mean, it's a really good idea. I think there's also on the flip side, these people have to live with these diseases their entire life. So if you could improve the quality, I mean, you could, strategically, I see your point, but you could make the argument either way. So I think what's really going to be important from the CRISPR perspective is pick a few solid cases that are going to work, so we can know if this works, so it doesn't cause problems down the road and perhaps hinder. Yeah.
Has CRISPR actually been used to cure someone who's had muscular dystrophy?
No.
Still research going on.
All research going on. The closest thing is that the T cell experiment that I mentioned that's going on underway at UPenn and also at Sichuan University. But no, this has never been done to my knowledge. I mean, maybe you know something that I don't know, but no. So I'm kind of running low on time. I should probably, I'll mention kind of germline editing therapy. You can imagine that, germline editing therapy. So now we're talking about changing the sequence of human genomes, right? And this is basically human. If there's a patient for Duchenne muscular dystrophy, you could ask them, do you want this therapy? Think about the risk. Do you want it? And they could say yes or no. If you're talking about an egg or a sperm, there's no patient buy-in, and there's an ethical risk, and that means, so basically it's kind of against their will in one sense. But also, if you're wrong, and if the effect of your therapy is worse than the disease itself, now not only do they have it, but their children have it, and their children's children have it, and their children's children have it. So there's a lot. And also sort of the slippery slope aspect. So what do we consider a disease? So there have been a few groups that have kind of written on this. Most recently, the National Academy of Science. This is sort of the federally chartered body of scientists that kind of weighed in and said that purposes other than treatment or prevention of disease and disability should not proceed at this time. But you can imagine a slippery slope case. So what is considered a disease or disability? So certainly muscular dystrophy, sickle-cell, clearly. But what about something like autism, which, in some cases to some people really is a disease that is debilitating, but others claim, to them it's just a different lifestyle. Now, if you can change it with autism, what about increasing intelligence or changing hair color? So it's easy to see where do we draw the line as to how this therapy becomes permissive. And this had been done, actually, from a technical perspective, being editing in human germline cells. It's been done a couple times. It was done in this lab in Guangzhou University. Basically used tripolar embryos. So these were cells that were fertilized by two sperms, so they're never possibly going to make a viable human, and they're basically trying to do CRISPR in the cells, and it worked to a medium extent. This was done in 2015 and led to a loud outcry of, well, what are we going to do? And kind of putting us at this Asilomar moment where I mentioned before, where people sort of need to come together and figure out a solution. And more recently, just recently, a couple weeks ago, that this basically was published. Scientists at Oregon Health and Science University took viable embryos and actually edited and did some kind of cool tricks, but showed that you could actually edit human embryos. And again, from a technical perspective, that's not really that shocking to me, nor is it to anyone else who kind of does this sort of thing. It works in virtually every embryo in which it's been done. The question is, how do you weigh the risks of that? So I think those are going to be some of the really big open questions. I'll give you. I'm sorry, I'm really going over here. I'm going to just show you kind of one more data slide. So I've got all about biomedicine. How many of you know the Sally Gardner at a Gallup movie, right? Yeah? You guys have seen this or something like it? And maybe you've seen this CRISPR thing already. This is one of the first motion pictures ever made, so this is before film, but basically just a series of photographs, and unfortunately I don't think it's animating. Is it animating? Oh yeah, good. Yeah. Nice. So here's the original image, downsampled a little bit, computerized. But using CRISPR, what a lab was actually able to do was to store that information, store that data in a bacterial genome, and then read that movie back out. So you're basically watching, like, E. coli Netflix here, right? I mean, the plot's a little kind of boring, and I think the character development's a little weak, but, it's pretty amazing, the idea how you could maybe, use CRISPR to re-engineer the way we think things that have really nothing to do with biology, storing data. So I think that's one really cool, interesting case. So this is kind of my wrap it up slide. So I'm going to start the music. Music's going to play me off soon. So I think in general, if we think I'm kind of therapeutic based, I guess, here. How should we proceed? Who do we make these decisions, and what standards should be used? And I think one of the things that's tricky is with Asilomar, there were a lot of people kind of all in a few countries that could kind of coordinate. I think one of the things that's really exciting about our scientific environment is that it really is dispersed throughout the world. And we might have our own culture associated with how medicine and what the standards should be, but not every culture is going to have the same standards. So how do we come up with sort of a consistent philosophy to govern what should and shouldn't be done. But in the meantime, we're using CRISPR all the time to learn about basic and biomedical research, other cool, exciting engineering enhancements, and CRISPR is just kind of getting online, and there's a lot of interest in making it even better. So, sorry, I'm over here, so feel free to scooch if you got, but I'm also happy to answer any questions.
George, you've been talking all night about therapeutic applications and possibly potential human applications. But of course, it's a general technology.
Yes. I didn't even get to agriculture and all these other,
and everything else.
Yeah.
There is, at least theoretically, an application called G drives.
Yeah, I didn't even touch that.
Where you can actually put sort of the mechanism into an organism, and it will go through and as it breeds throughout the population, it will modify the genes of every organism that it breeds with.
Yep.
Is there been any work, or we're talking about setting up standards, is there been work on setting up standards addressing that particular application?
There's an active group that sort of talked about it, published a paper. Professor Joe Wildonger here was an editor, an author on that paper. But if you guys aren't familiar with gene drive, as you nicely explained, probably better than I will. We tend to think of the way we think about gene inheritance is you have big A, little a. You might get big A or you might get little a. Now, what if you have big A, little a? Your little a change is a CRISPR change. What if little a can change big A into little a? So if you were big A and little a, now you're two little a's. So all of your offspring are little a. And if they mate with someone who is big A, now all of their progeny would be big A, little a. But now that CRISPR change changes them to little a, little a. So that's what's called gene drive. So you get this massive propagation of this version of the gene that just hyperdrives it. So the first case that's closest to being used, you can imagine, is there's a group that wants to use it to basically make mosquitoes immune towards some of it's amazing how many I think mosquitoes kill more humans than any other animal or something. Don't quote something. Mosquitoes can be really a problem because of the disease vectors they can act as. So the ability to take a mosquito and make it immune against a certain virus. You can imagine if you just took one mosquito and threw it out there, it would, maybe you would mate a couple times, or maybe it wouldn't. You'd hope it would, But if there's like a whole, rainforest full of other mosquitoes, that's a problem. So that's where gene drive comes in. So there is some hope to get this up and running and test it in a location in Florida, but for whatever reason, people didn't want that done. Didn't want, this massively promulgating version of the gene happening in their backyard. So it's not really clear logistically how this would happen, but it is a really promising aspect to CRISPR. And how to deal with it, I don't know. To some extent, you have to weigh the benefits and risks, but it's a great question. Yeah.
What agencies fund most of the CRISPR research, and what, in your opinion, is likely to be the impact of the Trump proposed cuts in scientific research? Implications of technology.
Sure. So National Institutes of Health are the big funder for most of the biomedical research. I'm sure there's USDA for the agriculture side, but NIH is funding a lot of the big bread and butter grants that use this research. And there are current NIH standards in place about editing human germline embryos. There aren't many more standards beyond that I'm aware of.
I'm not talking about standards, I'm talking about money.
Yeah, right. Certainly. So it's been brutal to be a publicly funded lab in general for a while now, and certainly cutting NIH budgets never help. I don't know that it's going to specifically affect CRISPR, but certainly I think one of the, if nothing else, what I think is going to cause it to occur is probably push everything towards research that's going to what's the thing that's going to most directly lead to a cure, right? And that's great, you know, from a practical side, that's great. If we only have so many grants to go around, what are the ones that are going to most directly benefit humans? But it's easy to imagine, you know, if that was strictly the case, we never would have learned about CRISPR. Because, I mean, the people that were studying bacterial genomes weren't trying to cure cancer looking at bacterial genomes. They're trying to understand the fundamental principles of biology. So I don't know that CRISPR itself is necessarily going to lose funding, but all the other things that could potentially lead to therapies, I mean, it's devastating. And frankly, you know, the public research becomes an incubator for other ideas that become IP. So, I mean, there are lots of other corporate strategies now where it's basically to check in on on publicly funded labs, and we have we have WARF on campus that's really good about making sure that the campus gets credit for the research that it makes. So, I mean, everyone loses, in my opinion, of course, is everyone loses when we don't fund science.
So, just to get this straight, CRISPR is not a piece of equipment, it's just a technique.
CRISPR.
It's done in the laboratory in little glass bottles.
Yeah, right. Well, that's funny. Exactly, because whenever people want to come and visit my lab, they're always waiting to see like the big billion-dollar CRISPR machine. Exactly, with all the like bright lighting and all the lever that I pull, like Willy Wonka or something.
And is E. coli the ink, is that used in almost all of the CRISPR things? Is that bacteria that you're using for?
So now it's like so abstracted. We almost don't even, from a personal perspective, I don't even deal with the bacterial nature of it. We've learned so much. There are lots of people that are studying it in its context and that will rely on them. But I don't use.
You always have to have that in the mix in order to break the.
Well, one of the nice things is that they learned it's been learned exactly which pieces you need. So you don't need to take like extracts of entire E. coli cells, for example. They know you need, and that's kind of the beauty of it, is that you only need a few components. That's really the other important thing. You only need that guide RNA, that's cheap to make. You only need that Cas9 protein, that's the same regardless of where you're targeting. So it's so simple, it's just a 2-component system, that's all you need. Yeah, exactly. But I probably should have a machine that has all kinds of flashing lights and smoke pouring out and stuff. Because people are always like, oh yeah, that's like my lab 15 years ago. Really? Okay.
Maybe you should just do it for show, and then since they're so excited,
yeah,
just show them the real thing now.
And then really let them down. All right, that's good. All right, I'll think about that. Sounds good. Any other questions? Oh, sorry, and then I'll dismiss you guys, sorry. Go ahead, yeah.
There's other things besides Cas9, aren't there?
Yes. Yeah, so, right, so, that kind of gets down to here, optimizations and enhancements of CRISPR-Cas9. So, the first one that, the workhorse is CRISPR-Cas9. It comes from this S. pyogenes species of bacteria, but there are more species of bacteria than there are, I don't know, there are a lot of species of bacteria, and they all have CRISPR. So there are a lot of people now combing through genomes of other bacteria, so there are other versions of Cas9, and they all cut it a little differently, and they all have their own different characteristics, and they all look for slightly different, they work at different, all kinds of different, they're all going to be a little bit different, so that's a tremendous, resource, the ability to comb through that. So Cas9 is the canonical one. It's like saying Kleenex, or like, it's a brand. I mean, it's one, but there's tons and tons of others. But you're right, Cas9 isn't the only one, if that's kind of what your question was. Okay, yeah.
One other observation, and that is that while it's a tremendous technical innovation, I think we should always keep in mind that ultimately depends on studying biodiversity. In other words, the bacteria are the ultimate genetic engineers. They came up with the entire system, essentially programmable restriction enzymes. And we've just improved it without preservation of biological diversity.
Sure.
You're going to have a much smaller range of things you can pursue.
Right. So if all you thought about in biomedical research is laden with that, you basically studied mice, or if you're interested in biomedicine, you studied mice, or you didn't study anything. So, and we need to have a more broad perspective about the different, there are strengths and benefits. So there's biodiversity from an ecological standpoint. Of course, I mean, you can't get by with just having reduced biodiversity. But even in the labs, what happens if we pin all this biomedical research on mice? Well, mice tumors aren't the same as human tumors. And in fact, many times there are rat tumors that look more like human tumors. And now with CRISPR, we can actually make genome-edited rats. So, I mean, you know, and in other cases, we work with swine because, you know, you can detect tumors in rats, and that's really useful. One of the important things for cancer is not just curing it, but also knowing is it there, detecting it efficiently. Well, I mean, how can you use this, if you invented a new piece of instrumentation, how can you detect cancer in a rat and then use the same piece of machine on a human? It's not really the same. So that's why we're looking to swine, where there are pigs that, you know, are virtually the same, have the same organ sizes, the same lifespan, same development, same tumor progressions. And so it really is important that we think about biodiversity, not just sort of in this usable way, which is of course really important, but also we not get one trick ponied, if that's a verb. But I feel like I should let you guys move about your day. So if you want to come up, thank you. Thanks.